How were major biochemical pathways discovered? MCAT Bio Biochem. Have you ever wondered how scientists figured out the complex web of biochemical pathways that keep our cells functioning? The story of these discoveries is fascinating and filled with creativity and hard work. Major biochemical pathways were uncovered gradually, mainly in the early to mid-20th century. This was a time when advancements in various fields like enzymology, chemistry, physiology, and structural biology came together. The first major pathway to be fully described was glycolysis. This process breaks down glucose to produce energy. Scientists like Otto Meyerhoff and his team made significant strides in the 1930s. They focused on muscle metabolism and fermentation. Through their experiments, they identified the sequence of reactions, the enzymes involved, and the intermediates that play a role in this pathway. Their work showed how energy is stored and released, particularly through adenosine triphosphate, which is now known as the universal energy currency of the cell. Two key trends helped in the discovery of glycolysis. First, scientists recognized that enzymes act as biological catalysts, driving metabolic reactions. Second, they identified phosphorylated compounds, such as creatine phosphate, which highlighted the role of phosphate groups in energy transfer. Advancements in enzyme chemistry were crucial. Researchers learned to isolate and characterize enzymes outside of living cells. For instance, the crystallization of enzymes like urease and catalase in the 1920s and 1930s proved that enzymes are indeed proteins. This opened the door for further studies on their structure and function. After glycolysis, other pathways emerged, including the Krebs cycle, also known as the citric acid cycle. This cycle revealed how cells extract energy from various nutrients through a series of cyclic reactions. By the mid-20th century, dozens of metabolic pathways had been documented and integrated into maps that illustrated their interconnections. Structural biology played a significant role as well. Starting in the late 1950s, scientists began solving the three-dimensional structures of enzymes. This allowed them to understand how enzymes catalyze reactions at the atomic level, providing clarity on biochemical mechanisms. For those preparing for the medical college admission test, understanding the history of these discoveries is beneficial. It helps to contextualize why metabolic pathways are organized the way they are and why enzymes function as they do. It also highlights the importance of enzymes and energy molecules like adenosine triphosphate in physiology and disease, which are essential concepts for medical students. In summary, the discovery of major biochemical pathways involved several key elements. Researchers identified enzymatic reactions and intermediates, beginning with glycolysis. They made advances in enzyme isolation and characterization, proving that enzymes are proteins. They integrated individual reactions into pathways that show energy flow and metabolic regulation. Structural studies revealed how enzymes work. Finally, they developed metabolic maps that illustrate how these pathways connect, which is essential for cellular function. This historical journey provides a solid foundation for understanding metabolism in medical education and the biological and biochemical sciences section of the medical college admission test.